Types of CA's enzymes and their functions. CRISPR-Cas systems are broadly classified into two main classes and further into 6 types, I6, and numerous subtypes based largely on the effector CA's proteins involved. Class 1 systems, types 1, 3, and IV. These systems typically involve multi-protein complexes for their effector function. Cas1 and Cas2. These are found in almost all CRISPR-Cas systems, both Class 1 and Class 2. Function. They are essential for spacer acquisition, the process where short fragments of foreign DNA, protospacers, are captured and integrated into the CRISPR array in the host genome. Cas1 is a metal-dependent nucleus integrase, and Cas2 often plays a non-enzymatic scaffolding role, forming a complex with Cas1 to facilitate DNA integration. Type 1 systems, for example Cas3, signature enzyme, Cas3, function. Cas3 is a multi-domain protein with both helicase and nucleus activity. It functions as part of a multi-protein complex called cascade, which unwinds and degrades foreign DNA after being guided by the crRNA. Type V systems, for example Cas12a/CPF1, signature enzyme Cas12a, also known as CPF1. Cas12a is an RNA-guided DNA endonuclease that generates staggered cuts with 5' overhangs and double-stranded DNA, unlike Cas9's blunt ends. It has a distinct PAM requirement and uses a single crRNA for guidance. No trace RNA needed. Cas12a has collateral activity, meaning it can cleave untargeted DNA once activated, making it useful for diagnostic applications. Other type V enzymes include Cas12b, Cas12c, each with unique properties. Type fixed systems, for example Cas13, signature enzyme Cas13. Cas13 enzymes are RNA-guided RNA nucleases. They target and cleave single-stranded RNA molecules. Many Cas13 enzymes exhibit collateral RNA cleavage activity, making them powerful tools for RNA diagnostics and gene regulation like RNA.
The CRISPR-Cas system is a revolutionary gene editing technology that originated from the adaptive immune system of bacteria and archaea. At its heart are the CA's CRISPR-associated enzymes, which play crucial roles in recognizing and cleaving foreign genetic material. Here's a breakdown of their structure, composition, types, and functions. Structure and composition of Cas enzymes. Cas enzymes are proteins, and their precise structure varies depending on the specific Cas protein. However, some general principles apply. Protein nature. All Cas enzymes are composed of amino acids folded into specific three-dimensional structures. Active sites. They possess active sites that facilitate their enzymatic activities, such as nuclease, DNA, RNA cutting activity. Many Cas nucleases require metal ions for their function. Domains. Cas proteins often have multiple domains that contribute to their various functions, such as DNA binding, RNA binding, and catalytic activity. For example, Cas9 has 2 nucleus domains, HNH and RUVC, which cleave different strands of DNA, guide RNA interaction. A defining feature of many Cas enzymes, particularly in Class 2 systems, is their association with the guide RNA, gRNA. This RNA molecule directs the Cas enzyme to its specific target sequence. The gRNA itself is a chimera of 2 naturally occurring RNAs in bacteria. CRISPR RNA, tRNA, and transactivating CRISPR RNA, tracRNA. TracRNA. The RNA acts as a structural scaffold for the Cas protein and contains the sequence complementary to the target DNA or RNA. Protospacer adjacent motif, PAM. Many CA's enzymes, like Cas9, require a specific short DNA sequence called a protospacer adjacent motif, PAM, adjacent to the target sequence for successful binding and cleavage. The PAM sequence varies between different Cas enzymes.
Type 3 systems, for example Cas10, signature enzyme Cas10. Function. Cas10 is often involved in targeting RNA, although some type 3 systems can also target DNA. They can have both nucleus and cyclic AMP-CAMP synthesizing activities, which can lead to a more widespread immune response. Type IV systems. Less well characterized, these also involve multi-protein complexes and are often found on plasmids. Class 2 systems, types 2, V and VI. These systems are characterized by single, large effector proteins that perform the target recognition and cleavage, making them particularly attractive for genome editing applications. Type 2 systems, for example Cas9. Signature enzyme, Cas9. Function, Cas9 is the most widely known and utilized CA's enzyme for gene editing. It is a dual RNA-guided DNA endonuclease. Guided by a single guide RNA, sgRNA, Cas9 introduces double-strand breaks, DSBs, at specific DNA target sites. The H and H domain cleaves the complementary strand, and the RUVC domain cleaves the non-complementary strand. This precise cutting allows for gene knockout, insertion, or modification through cellular DNA repair.